This is part 2 of our 8 fastidious gram-negative bacilli lectures. And in this lecture, we are going to focus on Haemophilus identification. So, one way to identify Haemophilus is through direct examination. And we mentioned direct examination in previous lectures. And what that is when you look directly at a clinical specimen. So in other words, you don't rely on growing the clinical specimen up on agar medium or in a broth. So you take that clinical specimen, usually you're going to put it onto a slide. Depending on what specimen it is, you would put it on a slide in various ways, and then you would gram stain it and then look for the organisms directly in the clinical specimen. That's especially done in cases of meningitis. We talked about that when we talked about Neisseria meningitidis, where you're going to commonly get a cerebral spinal fluid in, and that is immediately, it's a stat test, everything is dropped. You take your spinal fluid, you make a slide of it. Usually you're going to spin it down so that you get any bacteria and cells that might be there. You're going to get it concentrated. You're going to take that, whatever gets spun down, the concentrated what's called a pellet, you are going to make a slide, you're going to gram stain it and look and see what you have. So the Haemophilus in general are slender, gram-negative, usually coccobacilli, small, teeny tiny rods. They do not form spores, they are not motile. And here is your gram stain of a typical Haemophilus. And you could see it's very, very, very tiny. Some of the bacteria are a little bit bigger than others. Some are stuck together. When you see an individual Haemophilus organism, it's hard to tell if it's a rod or a cocci. Now, in a direct smear of a cerebral spinal fluid in the case of meningitis caused by Haemophilus influenza, you are going to see extracellular and intracellular gram-negative coccobacilli. And if you remember our Neisseria meningitidis and Neisseria gonorrhea, those coccobacilli, these don't look that much different. They do have that cocci look to them, so you have to be really, really careful. You can also see a little halo around these organisms, and that would be the capsule. Now, to cultivate these organisms, they are fastidious or fussy. Haemophilus or respiratory specimens. If you had a spinal fluid after you take some of the spinal fluid and do a direct gram stain on it, you would also do some platings, and you always want to plate on chocolate agar so that you can be sure that if Haemophilus influenza is there, it's going to grow. So the respiratory specimens are normally put in a 5 to 10% CO2 incubator. These organisms will grow pretty much anywhere. They can grow at lower oxygen levels. They like CO2. And growth is at body temperature at 35 to 37 degrees. Now, Haemophilus ducreyi are sexually transmitted infection, grows best at 33 degrees and requires special media called GC agar. And GC agar has vancomycin in it to reduce the growth of any normal flora that might be present. If you can remember, in our Neisseria gonorrhea lecture, we also talked about special media that contains antibiotics in it to reduce normal flora and enhance the growth of the Neisseria. So Haemophilus ducreyi has something similar and likes lower temperatures. You should get visible colonies of Haemophilus in 24 hours. They are usually somewhat transparent, smooth, convex. Haemophilus is a lot of times described as having a mousy odor or a bleach-like odor. Now, way back in the day, I mean way back in the day, even before my time, it was common in the clinical laboratory for laboratory scientists to sniff plates, and people with a lot of experience can just sniff a plate and know what was growing on it by what they smelled. Nowadays, we know a lot more, and sniffing of plates is not allowed at all. I mean, sniffing a plate can cost you to lose your job, possibly. It's just frowned upon. And that's because there are organisms out there like Neisseria meningitidis and many other organisms that you are risking your life by sniffing the organism growing. So you do not want to ever sniff a plate. However, textbooks still refer to the odor. When you open up a plate in the laboratory to do a catalase test or an oxidase test, sometimes the smell just comes at you and hits you, and you don't even actually have to sniff the plate. So we do still describe the odor of certain organisms if it's very characteristic. Now, encapsulated strains will produce a larger and more mucoid colony. Of course, that's common because capsule producing organisms usually are more mucousy looking. And the organism, if it's grown up in a broth, usually when you grow bacteria up in a broth, it gets very, very densely cloudy and turbid. And that is not as common with Haemophilus. And one of the reasons is because Haemophilus requires blood to grow, and most broths don't contain blood in them. So Haemophilus does not grow very well in most broths. We talked about the X and the V factors, or hemin and NAD, and how various species require either the X or the V, or X and V. Haemophilus influenza requires both X and V. So they have X and V discs. Usually you buy them in a disc. These are strips where you have your filter paper material that's impregnated with either the V factor or the X factor, or both X and V. And you'd make a lawn of your organism and drop your X disc, your V disc, and your X and your V disc, and you can see where the organism grows. If it requires V factor only, it will grow around the V factor, and it will probably grow around the X and the V because there's V there. But an organism that requires both X and V factor, like our pathogen Haemophilus influenza, is only going to grow around the disc that has both X and V in it. Now, of course, you would do this on a general supportive or nutrient agar plate that doesn't have your hemin or NAD, doesn't have any blood source in it. So you want to really see, is this organism only growing around the NAD and hemin that you are supplying it on that paper disc. Now, we already said that chocolate agar has both X because it's a blood source, so it has hemin, and it has the NAD because it is hemolyzed blood. So Haemophilus influenza, which requires both X and V factor, will grow very well on chocolate agar, and it usually produces a tan, somewhat mucoid colony. Now we're going to go over laboratory diagnosis. Now, we already talked about the X and the V factor, and what you can do is take 2 agar plates, one plate that contains X factor, like a sheep blood agar plate, and another plate that has neither X or V factor, just a basic supportive media like Mueller-Hinton agar. You could take your test organism, streak it for confluence, and add a V producing organism or a V disc and see what happens on the plates. So here for our X and V requirements, it looks like Mueller-Hinton agar, so no X or V is added at all. And you can put an X and a V disc somewhat close together. And if it requires only X, it'll grow around the X. If it requires only V, it'll grow all the way around the V. If it requires both X and V, it will grow in the area in between the X and V if they're close enough. The better way to do it is to use an X disc, a V disc, and a disc that contains both X and V. So on the left, this is Haemophilus influenza because it's growing only in between the X and the V strip, so it definitely requires both X and V factors. On the plate on the right, the organism is growing all the way around the V strip, so you know that this organism only requires V factor, and this organism is Haemophilus parainfluenza. You can do carbohydrate speciation tests, and if you remember in a previous lecture we talked about the rapid tests, and these rapid tests are little tiny kits that have little cupules, and you add your organism to it and you incubate them usually for about 2 to 4 hours, and based on the color reactions in these little couplets, usually there might be 8 or 10 different reactions in each one, in per test, and you come up with a little scheme, a number scheme, you put it into the computer and it'll give you the likely organism. Another nice easy way to differentiate your Haemophilus species is to use a quad plate. So this is an agar plate that has 4 different zones, and each zone has different media in it. One zone has media that contains only X factor, the other has only V factor, another quadrant has both X and V factors, and then the 4th quadrant has a media that has horse blood in it. So another test that used to be done in the clinical laboratory that's no longer done is the porphyrin test, and this test identifies the X factor requirement. And so what you would do is take a hemin-independent strain which produce porphobilinogen and porphyrins in the presence of delta ALA, or delta aminolevulinic acid. So you would take your bacteria and inoculate it into a delta ALA media and then expose it. You'd incubate it first, usually for about 4 hours, and then expose it to UV light. And so here's what you would get. Under ultraviolet light, the organism that is positive for a porphyrin reaction reacts to that delta ALA, and the medium will fluoresce. So the bottom organism is porphyrin positive, and the top organism is porphyrin negative. But this test, like I said, is not done in the clinical lab anymore. Quad plate, that's not commonly done anymore with your microscans and your Vitex. You don't need to do as many manual tests, but there are some labs that might do some quad plate verification tests, or if the instrument is giving inconclusive results and you're pretty sure it's a Haemophilus influenza, I would say that most labs are either going to have a default of a quad plate, or more likely a default of a kit test such as the rapid NH test. So we're going to now move on to the third section of the Fastidious gram-negative rod lecture.